Hi, this is Matt from Algae Research and Supply, and today we're going to try to do something a little bit different here. We have gotten a lot of requests from people to use the different colors of the rainbow, different wavelengths of light, to try to affect the rates at which photosynthesis takes place by giving the wavelengths of light that either will be more or less able to be grabbed onto by the photosynthetic pigments. So what we're going to do is we have one of our benchtop lights here. This is one of our algae lights that we manufacture. And from the fine people at Amazon, I picked up a whole bunch of different colored screens. These would be filters. If you're in theater, they would have been called a gel. I was a theater, well, not really a theater kid, but I did sing in a musical. I was a bad guy. Anyway, that's a story for another time. But what we're going to do is we're going to put these on the light here. But we don't really know much about how that filter works. So I have one of these things. This is a spectrophotometer or a spectrometer that has a little diffraction gradient inside there. And we're going to be able to use this diffraction gradient inside here to understand the spectrum or the quality of light, the wavelength of the light that's getting emitted by this LED light after it goes through these different filters. And we're going to compare that to the sunlight outside. And I think we're going to be able to use some AI software, mainly a Chat GPT program, to be able to look at that spectrum and to be able to actually make some graphs with it. At least we're going to give that a try. Finally, the one thing that we really want to do to make sure that we're giving the same amount of light, the total flux of photons at each individual one of these wavelengths here, we're going to use a spectro or a photometer. This is measuring PAR. These are from the fine people over at Vernier, and they have we have one of their little devices here. This is their Lab Quest. So we have the PAR meter here. So we're going to measure PAR, and PAR is it's a measurement of the flux of photons, or the amount of light that goes through, that's within the 400 to 700 nanometer range. So in between 400 to 700 here. And we'll be able to quantify that light, hopefully in a way that is going to be meaningful for us. So let's dive in and have a look. Okay, so we have this, which is the spectrometer, which has the diffraction gradient in it. And I have used all of these fine different colors here to get different wavelengths out and put them in front of our benchtop light as you see it. And then I've sat here like this to look straight down into it. And I have come up with these spectrums. Woo, spectrums, you can see them, all the different ones that are there. And from that, now we have the relative amount of photons which are coming in at which wavelength as a function of the filter. No, I'm not a cyborg. This is a PAR meter made by Apogee that is used by Vernier with this meter here. And this is a pretty cool little gadget. So what we're going to do is we're going to measure photons of light in between 400 and 700 nanometers. Down here, I'm just going to put this, I'm going to rig this onto this light stand, and I'm going to put the different wavelengths of light down here, or the filters on here, so that we can actually count the number of photons that make it through this filter here, so that way we know how much light is coming in PAR as a function of each one of these filters. And so we're going to put them all on there, find out which one has the smallest amount of light going through it, and then we're going to put on extra layers of filter for the ones that have a lot more light coming through. That way we can normalize it all to the minimum number of flux of photons through these screens. Lots of work, but it's really cool. It'll be worth it. You'll see. Yep, about 5 millimeters from the bottom. And the intensity of the reading that we're at, oh, it just went away. But that's how we're going to get it, so that we have the same spot, so that when I just change the screen here, we can go all the different screens in a row, and we can get an idea about what the flux of photons into the meter from that one specific LED are going to be. Now we have the red here that's got roughly 100 micromoles. So I'm going to go to the orange next and see how many layers we need there. Here's the 1. However, I think that the whole pack probably needs to be there to make this one work right. So let's go with this. And that's going to get us to 152. We're going to come back to orange because this obviously needs a lot of layers to get to 100. All right, so now we've made different layers of those light filters, and here's the screen now because that's our neutral density filter. We've got one layer of the red and then several layers of all of the other colors here so that we can go ahead and match out the total flux of photons by each color. And so I have gone ahead and used some packing tape to attach all of the light filters onto our benchtop light. We've got a number of different layers here depending upon which filter we used. The red only has a single one. Orange, yellow, the green, and the blue, and the screen all have different numbers. So I just laid them all out on there and we normalized them using the meter. And then I put a big piece of tape over all of them so they won't fall off as we do our stuff here. And now we will get going. Okay, so now we are getting ready to do the experiment. We have all of our colored filters there. We want to make sure that we don't have the algae bead stuck on the top. So shake those down so they're coming in contact with the light, or the exposure of the light. And we want to make sure that they are right over the LED so that we don't have anybody that is not getting less light. And from there, we'll check back in every 15 minutes until we see some color change. Let's see what happens. Oh, and just to make it all fair, I have this imported from Europe. This is aluminum foil. You can get this here in the States as aluminum foil, but it's lower quality if you buy aluminum. It's better to get the European version of the aluminum. It holds out the light, so it's a dark sample. Okay, all right, so here's our results. So we have the red here. These 4 represent the red. They definitely change to a nice shade of purple. Then we have our oranges here. Those also came into a nice purple. And then we start to see them go a little bit less dense in color. These 4 here are the yellow. And then we have the green that you can see these 2 have a little bit of a change and these pretty much none. The blue, I'm surprised as well. We only see a little bit of color change in the blue, and in the white we see less color change than we did in the red. I have a theory about that. But then finally we have all of our controls that were wrapped up in the aluminum, and I'll set those over here as well. And these are all, this is what everything started as, which is this fine shade of yellow there. So there's our dark controls. And again, we did see what we kind of expected here. My theory on why the bright light, or the white light, was less changed in color than the red, it's that it's spread across the entire spectrum. And there's a lot of things that are like here in the green and in the blue light that wasn't as significant for changing or for being responsible for photosynthesis. But the white light, we counted the total number of photons, which includes the green and blues. So I think that this is going to be a little bit of an average of all of them, as represented by the spectral output from the original spectrograms that we did. But anyway, here's the results. Okay, so let's sum this up. We took our algae light, or our benchtop light here, and we put on different filters, including a neutral density screen, to try to get a red, orange, yellow, green, blue, indigo, violet light, or just green and blue at the end there, so that we could try to find out which one of these colors, or how the quality of light or the wavelength of light by the color affects the rates of photosynthesis. And we have some data on that. I'm sure we could go and do more if we had some more time to do it. But the big takeaways, if you want to do this at your place, we don't have a product that has all these on it yet. We might, it depends on, if there's any demand for it. But the big takeaways for the DIYers out there are that each of these filters will attenuate light differently. This one doesn't reduce the total flux of photons as much as the blue one does. So we needed more of these than we did of these. And the reason that that's important is that we wanted to normalize how many photons of each of those individual colors got into the plant material, because we wanted to get everything over so that we had some color change. But we wanted to make sure that for each one of the colors, they all got the same number of photons. They got exposed the same amount of light. So that's why we had to go through these multiple steps. Not just that it's green light or that it's blue light, but that the flux of photons got in there the same way, or that the same number of photons got in there. So that is the entire project here. Please let us know if you have any other ideas or options, or if you found some errors that we made. I'm sure there are many in our assumptions about this experiment. Please let us know. We are scientists and we deal very well with feedback. We will not even kick you out of the press corps if you disagree with us. All right. Thanks so much, everybody. Take care. And because I promised, I figured I'd do this too. Okay, so I worked with ChatGPT up here and trying to figure this out, how to make it work. So there's the image that I uploaded, and I put all those on. It's a composite of all the different images that I made. And then I asked it to do it. I asked it to basically just do the red filter there, and it was incorporating all of these images, or all of the tick marks up there. So I had to iterate through it. And iteration is the secret to working with these computers. It did its stuff there, it did it again, and did it again. Finally, I got it to work by giving it this clip, which is only the red spectrum. And then I specifically told it, whatever you do, don't include the word red. And then it did. So that's there. So I know it's at least working, right? So I had to go then and later omit the word red, but it didn't want to admit the word red. It just wanted to scan it. So I took the word off, right? And then it finally did it right. So then I took the clue and I uploaded the whole thing and do them all. And it was like, oh, nope. So I had to go and change the image from this one. And then I made it into this one. I cleaned it up and I took the little tick marks out, but it didn't want that at all. It still made it really loud for the red. So finally I went down and cleaned it all up, took out all the words and everything at all. And then it came up with this graph, which actually looks pretty good. And so I'm gonna put that one on the end, that one right there, and that is that. I used AI to make this graph and it looks okay. I think it could be a little bit better.